Howdy and welcome to your unit on protein function. Now we're going to be doing protein function in two parts. Part 1 is the structure and function of HA, the influenza virus hemagglutinin spike protein. And part 2 is hemoglobin and myoglobin. Part 2 is actually covered in your textbook, but you'll have to dig further on in the chapters to find anything about influenza hemagglutinin. But what I like about starting your unit on protein function using hemagglutinin is that it harkens back to something that we learned before. It goes back to that heptad repeat of hydrophobic amino acids that we found in the primary structure of the fibrous proteins, alpha keratin. That coiled coil structure that we learned about before in the protein structure unit is applied in the protein function unit when we talk about how some viruses, bind to and enter your cells. And that's what I want to get to in this structure function of HA. HA stands for hemagglutinin, and hemagglutinin is on the surface of the influenza virus or the flu virus. As an introduction to the flu, I recommend that you watch this video. It's an animation produced by Prucell, who sells monoclonal antibodies against the flu. It's an approach that scientists are working on very hard right now, a similar approach for blocking entry of the coronavirus, which has a similar coiled coil structure in its spike protein. So what we learn about influenza virus is going to hold true for other enveloped viruses, that is, viruses that leave with a piece of your membrane around them. So these viruses have cellular membranes around them, and that's why they have these unusual and different looking shapes, because they're kind of squishy. They're not like a billiard ball like some viruses. They're envelope viruses, so they have membranes around them. And protruding off of the membranes are these spikes, and these spikes are the hemagglutinin, the HA protein. It's called something else in coronavirus and in HIV and in Ebola, but they all have similar structures to influenza virus. So this is an electron micrograph of what the flu virus looks like. When you zoom in to one of those spikes, and you look at just the membrane fusion portion. Now, what do I mean by membrane fusion portion? If you just look at this one spike protein, and then look at just the portion of that spike protein that actually is involved in viral entry into the host cell, then you see this cartoon. And on this cartoon, we're looking at a couple of things. We're looking at a transition from these loop regions here in yellow into a coiled coil structure, and also the delivery of these shorter alpha helices flanking the pink coiled coil in the middle to extend the coiled coil upward and carry with them this blue extended region of polypeptide at the amino terminus all the way up to the top of the molecule. The conformational change is 100 angstroms from here to here, and it was the largest conformational change known for a protein at that time. That is when I was a graduate student in the chem lab in 1993. I discovered this in 92, and it got published in Cell, and it got some publicity in the New York Times and the Washington Post in March of 1993. Okay. So what is the big deal about the function of HA, the influenza hemagglutinin? The big deal is the transition of regions that are not coiled coil into coiled coil in order to relocate the business end of the molecule to the top of the molecule where it can interact with the cellular membrane and stimulate the viral entry. So what are we talking about? We're talking about the cellular membrane is up here. The viral membrane is down here. You've got anchors into the membrane coming from each one of the monomers of the trimer. And then you've got these fusion peptides at the top in blue, which are actually doing the business of attaching to the host cell membrane. Okay, this would be the host, and this is the virus. All right, so that 100 angstrom or 10 nanometer conformational change is a random coil loop converting to a coiled coil and getting the protein ready to bring the host and the virus membranes together so that the virus can spill its nucleic acid into the host cell and make lots and lots of copies of itself. And that's what you'll see in the animation. This cartoon is one that I drew for Science Magazine right at the same time, well, a year later, when they wanted to publish our second paper on the subject. So what you're looking at is a kind of zoomed out view of what's going on. So now the pink coiled coil region in the prefusion, the left hand cartoon over here, this part over here is this part right here. And you see there are some green balloons, one for each one of the pink monomers of the homotrimer. And those are the receptor binding regions, and they bind to a receptor on the host cell membrane, which is this blue, and then they get endocytosed by receptor-mediated endocytosis into an endosome, where you see the conformational change that we were talking about, and there's the viral nucleic acid exiting out of the virus into the cell for replication. Okay, so the paper that published the structure, the atomic resolution structure of the hemagglutinin, came out a year later in Don Wiley's lab by Per Bulla and my friend Fred Hewson over in Harvard when I was at MIT. And the great part of the story, there's a couple of really fun things that happened here. Is first of all, I was studying coiled coils, working with my friend Brad Stewart, who was helping me optimize an algorithm to separate generic amphipathic helices from bona fide coiled coil heptad repeats of hydrophobic amino acids. And we were using a training and a test set, and hemagglutinin was in the training set. And so we knew it had that coiled coil, but when Brad pointed out that the coiled coil was actually the highest scoring region was this yellow region here, I told him I was going to have to check his math. So I did it by hand. I calculated the propensity for this region to form a coiled coil. And sure enough, this had a higher score than the known coiled coil, which led to the model for this conformational change, which I was able to prove spectroscopically with circular dichroism spectroscopy and analytical ultracentrifugation to show this region forms a trimer and a coiled coil. And that's what was published in Cell in 1993, if you're interested in seeing that. Okay, so the atomic resolution structure of a monomer of this conformational change came out the next year when Fred had been sending me papers through his wife, Liz Gavis, who actually worked right across the hall from me in the Whitehead Institute. So this was before the internet, and, well, we had internet, but it was all intranet at MIT. We could write to each other and exchange things, but there was no, World Wide Web, it was called at that time. It was about a year later. So what we did instead was I would find references to papers that I needed to read, send those references to lunch with Liz. She would have lunch with her husband, Fred, and then she'd come back from lunch the next day with the papers that I requested the previous day. And Fred got very jazzed and excited about this model, and he and Per Bulla solved the structure, and he called me on the phone a year later and said, "You were right. We have the structure, and it is an extended coiled coil." What they also found was that another region of the protein, a region that had been in that pink region, which is now yellow plus blue, actually breaks and bends back up. So what you have in the end is a conformational change where the pink region, in the context of the entire trimer, is a coiled coil before fusion, The yellow region is that loop region that had a high prediction of coil-coil structure, and the red is that outside small alpha helix, and the blue was the fusion peptide that acts like a hairpin anchor over here. And then after the conformational change, you can see that that yellow region is now in that included in that coil, as is the red alpha helix. So this was the structure that Per Bulla and Fred Hewson solved in Don Wiley's lab in 1994. And then the break and the bend back up is what they saw over here, which had a consequence for the mechanism of membrane fusion, the fusion of the viral membrane and the cellular membrane, remember, that is required in order to empty out the viral nucleic acid into the host cell. And I didn't show it, but it basically, if you take this molecule and turn it on its side, you're pinning the two membranes together and allowing those membranes to fuse. Okay, so there are not only similarities in the morphology of the flu virus, the Ebola virus, and the coronavirus, which you've seen lots of pictures of. They all have those trimers that are structurally and functionally similar to the HA, the flu virus hemagglutinin. The spike proteins are seen on Ebola virus, and they're even longer on coronavirus. The coronavirus has long and skinny spikes, but they're working the same way. That is, these helical groups rearrange to extend a coil to throw a hydrophobic anchor into the target membrane to then pull the target membrane back and fuse the viral and the cellular membrane. So they're all working the same way. And here's a structure in a space-filling model of the Ebola virus trimer on the surface of the Ebola virus membrane in gray. So that's just one example. And if you want to see the structure of the coronavirus spike protein, I've posted that paper in the folder for the viral membrane fusion proteins. This part 1 of our protein function. So in summary, what I want you to take home from this lesson on protein function, where we apply what we've known about the heptad repeat of hydrophobic amino acids in the primary structure of coiled coils, in the coiled coils of enveloped virus fusion proteins. I want you to learn that membrane fusion is exergonic. No ATP is required. Membranes fuse together and they have pretty much the same energy as they did before they fused. And so it's very slow, because that would be a bad thing. If all of our membranes were fusing, we would have one giant cell with no organelles, right? So we need special fusion proteins to overcome the barriers that keep our membranes apart. So those are called fusion proteins. The membrane fusion proteins undergo a conformational change that results in hydrophobic or amphipathic sequences inserting into both of the fusing membranes. And we'll see this again when we talk about membrane biology and biochemistry in chapter 9. We'll see that in the cell, the transport proteins that move transport vesicles from one compartment in the cell to another also use this strategy to fuse those transport vesicles to their target compartment. And in that case, and in the case of viruses, the fusogenic conformation of membrane fusion proteins are these alpha helical bundles between membranes that pierce and pull the membranes together. They pierce by putting a membrane anchor into the host cell membrane, and they already have a membrane anchor in the viral cell membrane, and then they rearrange into those coiled coils that bend over, and that rearrangement is what actually pulls the membranes together. In the next video, we'll talk about myoglobin and hemoglobin.